Welcome back to Chapter 12, Part 3. In this part of my lecture, I'm going to be discussing chromosomal aberrations, or also known as alterations, and I'll use those two terms interchangeably. So if there is a very large scale chromosomal alteration or aberration in humans, and in other mammals too experience this often leads to something called a spontaneous abortion, which is most people refer to as a miscarriage, but the proper biological term for this is spontaneous abortion. It can also result in a variety, several different kinds of developmental disorders, which we'll talk about some of them. Interestingly, plants can tolerate much more of these kinds of chromosomal alterations that I'll be talking about with you, and better than animals do. But we're not going to be talking about plants too much. It's just something that's really cool about plants, and I'll mention that again briefly in a second. So, what are these chromosomal alterations? Well, you can have a change in the number of chromosomes, or you can have a large-scale change in the structure of the chromosomes. And so we're going to go into details on both changes in number and structure in this part of the lecture. So let's start out with having an abnormal number of chromosomes. This happens in a process called nondisjunction. This is such an interesting word, nondisjunction, because it's got like a double negative in it. Non is a negative and dis is a negative, so nondisjunction. But basically, it's saying that disjunction is normal and that nondisjunction would be not normal. So in normal meiosis, you would separate homologous chromosomes. But in nondisjunction, what happens is that one of those chromosomes, one or more of those chromosomes, does not separate properly in meiosis. This can also happen in mitosis. It can also happen in meiosis 2. So I'm showing you here a nondisjunction in meiosis 1. And what results in that is that eventually, if you go through meiosis 2, the 1, 2, 3 chromosomes that ended up in this, they all separate, their sister chromatids all separate, and you end up having n plus 1 number of chromosomes, where n is the haploid number. So you should have ended up having, let's see, if we look on this one, you should have ended up having 2 chromosomes, because you got a total of 4 in the diploid, you should have 2 in the haploid, and that didn't happen because of nondisjunction. You have n plus 1, or 3 chromosomes, and that's going to happen in these two cells. In these two cells that ended up not getting the homologous chromosome, you have n minus 1, or in this case only 1 chromosome. Again, this can happen in meiosis 2. It can also happen in mitosis. So here's meiosis 1 going along just fine. But in meiosis 2, the sister chromatids didn't separate properly in this cell. You can see that they did separate properly in this cell. So these two cells here are normal, and they have n number of chromosomes, or 2 in the case of this example. But this one didn't separate properly during meiosis 2. I have to tell you that it will later be forced to separate, because eventually this cell is going to go through another round of mitosis, for example. And so as long as those eventually do separate, we have n plus 1 number of chromosomes here, and we have n minus 1 number of chromosomes here. So the result here is that one gamete is either receiving more or less chromosomes, and that's a huge problem. If you have less chromosomes than you're expecting, then there's some information that's going to be missing. If you have more chromosomes than you're expecting, then you're going to have too much information. And it turns out, at least in humans and other mammals, that having too much information is a bad thing. I don't normally show you videos in my lectures, but I did want to show you this one because it's very interesting. So if you look at this, this is actually a cell in mitosis, and here they are all lining up on the metaphase plate right there, right? You can kind of see them lining up on the metaphase plate, except for this one right here, right? And that one right there didn't separate, but there it just separated just there. So it's like this is on a loop, so you can see that it's going to show up. I think they do it one more time. Yeah, here we go. So it's just not showing up in on the other side, and that is a nondisjunction. When nondisjunction happens there it is right there. When nondisjunctions happen in mitosis in a fully developed organism, and there it is right over there, then what happens is that that can frequently lead to cancer. So that's nondisjunction. Nondisjunction is the act of being separated wrong, right? Correct? It's separating wrong. But the result of nondisjunction is called aneuploidy, and that's this word right here, aneuploidy. And to be euploid means to be true, have a true number of chromosomes. So aneuploidy means to not have the true number of chromosomes. Aneuploidy is referred only to after fertilization happens, where two gametes fuse, and which one of them has undergone nondisjunction. So if we really do look at the zygote here, and offspring that have aneuploidy are going to have an abnormal number of a particular chromosome, whichever chromosome didn't go through the disjunction properly, or you could also say it went through non-disjunction. So in this zygote, here's an egg that has n+1 chromosomes. Here's a sperm cell that has the normal 2. And you can see here that for this chromosome right here, the larger of the two chromosomes, that actually has 3 chromosomes in it, and that is aneuploidy. If we look at it in humans with all of our 46 chromosomes, we can see here are 2 different examples of aneuploidy. This one has an extra chromosome number 18, and having an extra chromosome number 18 often results in spontaneous abortion. It can also result in a child being born with Edwards syndrome, and that child, generally speaking, dies before age 2. Another example is seen right here of aneuploidy, and this one is missing a chromosome, and that's chromosome number 7 that's missing. If you're missing chromosome number 7 after a fertilization, if a zygote is missing chromosome number 7, that will absolutely result in a spontaneous abortion. So we can categorize these abnormal chromosome numbers with a couple of words. The first word is monosomatic, and a monosomatic zygote has only one copy of a particular chromosome. So this one right here, this zygote, is monosomatic. Or you could be trisomatic or trisomic. Sorry, it's monosomic and trisomic. I was just pronouncing those words wrong. You could be trisomic, like we see here with chromosome 18, that ends up with something called Edwards syndrome. Again, trisomic, the zygote has 3 copies of a particular chromosome. We'll talk more about that later, but I did want to introduce to you now the concept of polyploidy. Polyploidy is a condition where an organism has 2 or more complete sets of chromosomes. Unlike when we were talking about here with monosomy or trisomy, when we talk about polyploidy, it is a complete set. So you can think about the haploid number, that's like when you make gametes, right? And the diploid number, we consider that to be the normal number. But if you don't do things right, or if you have a haploid gamete fusing with a diploid gamete, which can happen in plants, you would have a triploid offspring. Or if you have 2 gametes that are both diploid, you could end up with a tetraploid zygote. And again, that sounds really weird to us, but because it just doesn't happen in humans, and most mammals, this does not happen. But it is fairly common in plants. This is how new plant species evolve. I mean, it's not uncommon for it to happen, and if you do this, then you get a new species of plants. That's basically because we consider plants, species, to be based on how many chromosomes they have. So spontaneous origin of polyploid individuals does play an important role in evolution of plants in particular. Again, not so much with humans and mammals and things along those lines. Besides having whole chromosome issues, we can have alterations of chromosome structure, right? So instead of counting entire chromosome numbers being off or being doubled or tripled, we can just have little bits of whole chromosomes having things happen to them. We have actually 4 major changes that can happen to chromosome structure, including a deletion, where part of that chromosome, a big chunk of that chromosome, goes missing. You can have duplication, and this sounds kind of weird, where a part of a chromosome replicates itself, and only part of that chromosome, so we say that like a segment of it is repeated. You can have an inversion, where the original order of the letters in the DNA sequence are reversed, and that happens within a chromosome. And then you can have a translocation, and a translocation takes a bit of one chromosome and puts it on a completely different chromosome. Let's take a look at what this means kind of more visually. So here is an example of a deletion, where you have the sequence A, B, C, D, E, F, G, H, right? These are different sections of the chromosome, and a deletion would remove section D, and so now our chromosome is shorter and is missing a huge amount of information. Deletions are pretty bad things to happen. You can have a duplication, and with duplications, you have the sections repeat themselves. So instead of just being A, B, C, D, E, and so forth, you would have A, B, C, B, C, D, E. And you might think that that would be a bad thing as well, because too much information can be a bad thing. On the other hand, duplications give rise to interesting changes into body types. We know, like, our spine is made up of a whole bunch of different bones, and that's in part resulting from a bunch of duplication events that have happened in the genes that lead to those bones. So duplications don't have to be bad. The next one is an inversion. So whatever the sequence of genes were on that chromosome A, B, C, D, E, those get flip-flopped around. A, D, C, B, that section right there got flipped, and then the rest of it looks normal. So that's an inversion. Finally, we have the translocation. And with translocation, you have to be talking about 2 different chromosomes. So we've got this one that we've been looking at so far, A, B, C, D, E, F, G, H, and we've got another chromosome here that has sections M, N, O, P, Q, R. With a translocation, the sections here in the darker color are going to switch which chromosome they're on. And this is called, again, a translocation, but specifically, this is called a reciprocal translocation because it's happening with both chromosomes, or they're basically trading. You can have non-reciprocal translocations, which you see that word right here. In that case, one chromosome would transfer its information to another chromosome, but the other one wouldn't give it back. So it wouldn't be an even trade. It would be an uneven trade. Let's take a look at things in another way here. If you are a diploid embryo and you are homozygous for a large deletion, it's likely that you're missing a huge amount of important genes, and that would be lethal. And so that would result in a spontaneous abortion. Duplications and translocations do tend to be harmful, but don't necessarily end in spontaneous abortions. And inversions, if you have the right genes there, then as long as they're there, does it really matter what chromosome they're on? Well, it might matter what chromosome they're on. It might influence it, but it doesn't necessarily have to influence it. And so inversions are, of all the ones that we've talked about so far, inversions are the least deadly type of alteration in a chromosomal structure. What we're going to spend the rest of this lecture on is just talking about a few, not all of them, but just a couple human disorders that are due to chromosomal alterations. And again, we're talking about either altering the chromosome number or the chromosome structure. And so again, we're going to look at like aneuploidy and what happens to individuals with a particular kind of aneuploidy. We'll look at a couple of them. And then we'll look at what happens to an organism that, a human that has had some sort of chromosomal abnormality with regard to structure. So the first thing that we're looking at here is Down syndrome, and this little girl has Down syndrome, and this is an aneuploid condition for chromosome number 21. So here's chromosome number 21 right here, and there are 3 chromosomes there. So that's also a trisomy. And so another name for Down syndrome is trisomy 21. This is a fairly common genetic disorder. It affects about 1 in every 830 children in the United States. We do know that the frequency of Down syndrome increases with the age of the mother, but we don't know why that happens. And there's been some hypotheses out there, but what we think is that there is an age dependency in humans or how females make our eggs, and there's this idea that women or female children are born with all the eggs that they'll ever make in their life. When they're born, there's some mixed information about that. So for now, we're going to put down with we don't really know the reason for why this has happened, but we're having some pretty good ideas about it. For men make sperm throughout their lifetime, and so they're less likely to have this kind of trisomy event happening or nondisjunction event happening. Aneuploidy of sex chromosomes is fairly common, and I'm listing 4 of them here for you. 2 of the 4 result in something that is considered a syndrome, meaning that it shows up and it appears to have some effects on those individuals who have the aneuploidy, but it doesn't necessarily have to. What we do know is that aneuploid for sex chromosomes is less deadly than aneuploid for the autosomes or the non-sex chromosomes. So here are the 4. The first one is Klinefelter syndrome. And Klinefelter syndrome results in having an X, it's a male. Here's the chromosome, here's the aneuploidy right here. They have 2 X chromosomes and 1 Y chromosome. And so this person's male because they have the Y chromosome, but they do have that extra X chromosome, and it does result in some physical differences amongst men who have Klinefelter syndrome. The next one that we're going to talk about is extra Y. So about 1 in 1,000 males are born with having an extra Y, and that would be XYY. This is a trisomy, but it turns out that having an extra Y chromosome doesn't seem to have any effect on males who have it. And in fact, remember that the Y chromosome is pretty small and doesn't have a lot of information on it. So that makes sense that having an extra Y chromosome might not be too disruptive. Females can have trisomy X, and their genetic complement would be XXX. if a female is trisomy X, they don't have too much differences. It's really hard to tell. I mean, just by looking at somebody, you couldn't tell if they're trisomy X. On average, they may be slightly taller, but really, it falls within the normal range. So all three of these have been trisomies. This last one is a monosomy, and monosomy X is called Turner syndrome. And the way that we refer to if a chromosome is missing is we use a 0 as a placeholder. So we would say that somebody with Turner syndrome is XO. So females with Turner syndrome do tend to be sterile. It's the only known monosomy in humans. And you might be asking yourself, why is it that we have 4 different aneuploidies of sex chromosomes, but we really don't know very many other aneuploidies besides Down syndrome that exist in humans? And this goes back to the concept of the Barr body. Remember that in humans and other mammals, X chromosomes can be inactivated. This is not true for other chromosomes. This is only true for the X chromosome. And so if you're a female with trisomy X, you end up inactivating 2 of those chromosomes. And it's not perfect inactivation, so there can be some difficulties with it. But again, if you're a male with Klinefelter syndrome, one of those X chromosomes can be inactivated in a Barr body. And so you generally live a perfectly normal life, with a few exceptions that happen because, again, the X chromosome inactivation is not perfect. The last thing that we're going to talk about is a structural abnormality. And in this case, we're talking about a structural abnormality called Kudushot. This is French for cry of the cat. And it's called Kudushot because children born with Kudushot are not able to speak in a normal voice. Their voices sound like a cat crying, so that's why it's called Kudushot. So Kudushot results from a deletion, a very specific deletion off the end of chromosome 5. And a child born with the syndrome is just severely intellectually disabled, has that cat-like cry that I was talking about, and they tend to die in infancy or early childhood. So having that deletion is a very, very harmful thing. Also, and this is the very last thing that we'll talk about, that there are certain cancers that are caused by translocations. In this case, we're going to talk about chronic myelogenous leukemia, or CML. And this is caused by a translocation. It's a reciprocal translocation where chromosome 9 and chromosome 22 trade little bits of information. And so, again, you'd think that that would be okay because the information is there, but it does end up having this form of leukemia. And a chromosome that has a reciprocal translocation there, like we're talking about, is often referred to as a Philadelphia chromosome. I can't tell you why it's referred to a Philadelphia chromosome, but that is something that you might hear. Well, that's all that I have for you on chromosomal inheritance, and I hope that this was informational, and let me know, as always, if you have any questions.